right? So what I'm interested in you understanding and knowing is identify the different elements here, so the different elements of homeostatic control mechanisms here, all right? So remember that we have a stimulus, sensory receptors are going to detect the stimulus and convert the stimulus into an electrical signal. This goes via the afferent pathway to the central nervous system, right? And that's what you have here. So you have receptors. The stimulus is not here, so that's another thing that I told you already a couple of times, but I'm not going to show, sometimes I'm not going to show all the elements in the diagrams, all right? Some of these elements are implicit, but I'm not going to show them explicitly. I'm not going to make boxes for different things, okay? So the stimulus is not here, but the stimulus is presence of food in the stomach, right? So this food is going to be, presence of food is going to be detected by the sensory receptors. This is going to be translated into an electrical signal, which gets to the central nervous system. And then the central nervous system is going to use the efferent pathway, right? Now, in this case, we're talking about glandular secretions, right? So the branch of the efferent pathway that we're going to use is what branch of the efferent pathway we're going to use? The enteric nervous system, and there is also the parasympathetic nervous system playing a role here. So both are going to send messages from the central nervous system to the effectors, which in this case are going to be stomach glands, right? But this is a very complicated diagram, right? There are very complicated interactions between all these things. So we're going to have multiple effectors and multiple responses, right? So we're going to have first let's say the cells that secrete hydrochloric acid, all right? So you have, those are going to be exocrine glands. Remember that the cells that secrete hydrochloric acid, so they form unicellular glands. These cells are called, the name of the cells that secrete hydrochloric acid is chief cells. Then the response of the exocrine glands in response to the messages that are sent from the enteric nervous system and the parasympathetic nervous system is to secrete hydrochloric acid, right? So that would be an effector and a response. But you can see that the hydrochloric acid also stimulates, for example, the secretion of somatostatin, all right? But somatostatin is a hormone. So hydrochloric acid is going to be kind of the signal for another effector, and in this case, since this is a hormone, the effector would be an endocrine gland, okay? To secrete somatostatin, let's say. Somatostatin, you can see that inhibits the secretion of gastrin and the secretion of histamine, right? So somatostatin is going to affect two effectors. Those two, gastrin and histamine, are hormones as well. So somatostatin is going to affect other endocrine glands and another endocrine gland. Each one is different kinds of glands, but both are going to secrete hormones. This one is going to secrete gastrin, and this one is going to secrete histamine. You can see that histamine affects the secretion of hydrochloric acid. somatostatin affects the secretion of hydrochloric acid, gastrin too. So this is going to affect this. This is going to affect this. This is going to affect this. So you have multiple levels of effectors and responses, right? So that can be like really complicated. I didn't include everything that is going on in the stomach here. So this is just a few molecules that we secrete that affect the stomach secretions, okay? But you can reduce this, so if you want to make this simple, so what you can say is that this is effectors, responses, right? And then you can complicate that as much as you want. So do not be intimidated by this, because you can make a complicated diagram, but I want you to understand that this can be reduced to that, okay? So if you understand the relationship between all these elements, then you're gonna be fine, okay? Now, all these things, the secretion of hydrochloric acid, the secretion of somatostatin regulating this, gastrin regulating that, histamine, blah, 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 all these things, each one of the factors has a specific response, right? So this endocrine gland, the specific response of this endocrine gland is the secretion of somatostatin. The specific response of the chief cells is to secrete hydrochloric acid, all right? And then you can see that this is very complicated and all these glands interact with each other. What's the overall response? So if you want to reduce all this to that, what would you write down? So the effectors are going to be stomach glands, right? So if you want to summarize all this, you can see that here each one of the effectors is a gland, right? Exocrine or endocrine glands of different kinds. So overall, in summary, the effectors in this case are stomach glands, right? Which is what I wrote that down there. And what would be the overall response? Here you have specific responses. Secretion of hydrochloric acid, secretion of somatostatin. Why do we do all this? To digest food. So the general response would be digestion of food in the stomach, right? Now, this is about secretions, so this specifically would be what kind of digestion? Remember that there are two kinds, right? Chemical digestion of food. All right? Do you see that? So this is the very general thing. This is in all homeostatic control mechanisms. You have all these elements and it ends up in effectors and responses, right? Specifically for stomach secretions, you have these effectors and this general response. And now you can break down all these into many different elements, all right? So realize that there are multiple levels of response, but overall, the main concept of what homeostatic control mechanisms do and how the elements are organized is pretty much the same, okay? Is that clear? So you're going to have plenty of chances to understand that because there are going to be more to come. All right, so this is how we regulate the stomach secretions. Now remember that the stomach does, out of these four processes, digestion, secretion, motility, and absorption, the stomach does two of them heavily, secretion and motility. And as a result of these two, it does digestion, right? And there is very little absorption, so we're not going to talk about absorption in the stomach. We started with secretion, now I'm going to continue with motility. Those are the two main processes that happen in the stomach. And remember when I talked about the esophagus? So how we move the food from the mouth to the stomach? We use a mechanism that is called, do you remember? Peristalsis. Good. And in peristalsis, you have a wave of contraction, okay? That of the muscles of the muscular sternum. So you have the wall of the esophagus, right? And then if you have the food coming from the mouth, if you contract these muscles here, then what you're doing is basically push the food in this direction, right? So if you can generate a wave of contraction that does this, and you can coordinate which are the muscles that you want to contract at each particular time, then you're going to move the food from here to here, right? Well, in the stomach, the same kind of movement happens. We have peristalsis. This is to illustrate that, these two first pictures. So here you have the esophagus, and here you have the small intestine, right? And this is the stomach. Now, when the food gets from the esophagus to the stomach, so the stomach is much larger than the esophagus, right? But it's part of the tube, okay? And then you have the small intestine here. When the food arrives finally here, the food particles are still pretty big, right? And then we need to digest them, so break them down into smaller components that then the small intestine can absorb, right? And we need to break down the food items, and we need to send them to the small intestine, all right? So with peristalsis, you have the same mechanism than here. So you have a contraction of, let's say, this part of the stomach, all right? So then you will have something like this. This contracts, and when the stomach contracts, what's going to happen? The food is going to be pushed down, right? Then the next step would be relaxation of these. Just going to draw the stomach because you have the esophagus, and it would be relaxation of this part of the stomach. Let's see. So now this region of the stomach is relaxed, and we contract the next region of the stomach, right? Now, if the food was here, now the food is going to be pushed here, right? Then at some point, you're going to contract this region of the stomach, right? You are producing a wave of contraction, and eventually you're going to contract this region of the stomach, and now the food can go to the small intestine, right? While the food is in the stomach, is going to the wall of the stomach is going to secrete all these things that we talked about right before. So then the food is going to be mixed with hydrochloric acid, with pepsin, with other enzymes, okay? And the food is going to be broken down. How long do you think it takes for you to swallow a piece of whatever? So you're eating an apple, and you swallow the piece of or the granola bar. How long, when you swallow what you chew, okay? How long do you think it takes to get the food from the esophagus to the stomach? Guess. How long? Seconds. It's pretty fast. Now, I'm telling you that the stomach is doing the same thing. It's producing peristaltic waves. So, how long do you think the food will take to move from the stomach to the small intestine if we use peristaltic waves? And the stomach is more or less the same length as the esophagus. So how long do you think it will take? Seconds. Are we interested in that? or are we interested in leaving the food in the stomach for a much longer time? We want to leave the food in the stomach for a much longer time because with, let's say, 5 seconds or 10 seconds, these pieces of food, they don't have time to mix properly with all the enzymes and all the hydrochloric acid, and they are not going to be broken down into components that we can absorb. Okay? So, how do we prevent these to happen, this really quick transit of the food through the stomach? How do we prevent that? Well, the difference between, so at the entrance between the esophagus and the stomach, there is nothing here that blocks the transit of food. But here we have a sphincter. And a sphincter is a ring of muscle, and this is not open like that. A sphincter is a ring of muscle that actually acts like a gate, right? And the sphincters can be open or closed, bless you. They can be open or closed, right? If they are open, obviously, things can go through them. If they are closed, things don't go through them. This sphincter that is located in between the stomach and the small intestine is what we call the pyloric sphincter. Remember that name? So when we talk about the general anatomy of the stomach, so I told you that there are different regions of the stomach, the fundus, the body, etc. So, but we don't care about those. So those are just like anatomical regions. But this one here, the pyloric sphincter, is important. Now, when a first wave of contractions is generated in the stomach, the food goes through there. But then whenever the food gets here and these muscles are contracted, the pyloric sphincter is going to be almost completely closed. There's going to be a tiny orifice there, all right? So this orifice, this hole, or an orifice is a hole. So this tiny, tiny hole is big enough for very small molecules to go through. So if you have a big chunk of a granola bar that gets to your stomach, the acids and the enzymes are going to break it down. And they're going to break it down into glucose, amino acids, small lipids, okay? But there are going to be chunks of the food that they didn't have time to get digested, right? So, but part of the food is already digested, a very small portion, so you have like different portions of food here. Probably all these things in the surface, they're absolutely exposed to hydrochloric acid and enzymes, so probably tiny molecules are already broken down, okay? Now, these small molecules can go through the pyloric sphincter, but the bigger chunks of food, they are too big and they cannot cross the pyloric sphincter. So, food items that are too large to go to the small intestine, they are going to be deterred here. And then what happens is that another wave of contraction is going to happen, but the food that was accumulated here that cannot cross the pyloric sphincter is pushed back. All right? So if the food cannot go to the small intestine, eventually this food, so you contract this and relax it, then the food is going to be bounced back. All right? And then you start a new wave of contraction, and these food items are going to get another round of mixing with the enzymes, the hydrochloric acid, and they're going to be smaller. If they're not small enough yet to go through the pyloric sphincter, they're going to be bounced back again, and so on, until they are completely digested. And then when you have pretty much all the food items are broken down into their components, into glucose, amino acids, etc., then everything can go through the pyloric sphincter and everything ends up in the small intestine. But now you're going to have things that are ready to be absorbed. Okay? So these movements of the stomach, they have basically two functions. So the movement itself grinds the food, right? The contractions of the stomach are pretty strong. And yeah, when you contract these muscles, remember that the stomach has 3 layers of muscle instead of 2, so the muscularis externa is very well developed. These contractions are really strong, and then they're going to crush the food items, okay? So they're going to grind the food items. And at the same time, the pyloric sphincter, since it's almost closed, will bounce back all the food, and then you have more time for the food to mix with the enzymes and the hydrochloric acid. And at the same time, the food that is already digested can move towards the small intestine, right? Is that clear? So, this tiny difference, presence of an sphincter here, but not here, it makes a big difference in the overall process, right? Without the pyloric sphincter, you will send big food items to the small intestine. The small intestine is not going to be able to absorb all this, and moreover, you can damage the wall of the intestine because these items might be like really big, right? Okay. So in here you have a first wave of contraction. You can see that the food goes from here to here, right? But then the pyloric sphincter stops the big food items to cross, and then the big food items, so they bounce back, they're brought back to the body of the stomach. And only the very small particles can cross the pyloric sphincter, and then they can go to the small intestine, right? How do we regulate that? So here you have a diagram here. And again, you should be able to identify the elements of homeostatic control mechanisms here, but I'm going to help you a bit because there is a twist here. So you can see that we start with the receptors at the small intestine, right? So remember that to regulate the stomach secretions, we start at receptors that are located in the wall of the stomach that can detect the presence of food in the stomach. But here we are talking about receptors that are located in the walls of the small intestine, okay? So we have receptors for osmolarity, we have receptors for hydrochloric acid, for acid basically. We have receptors that can detect fat. So when you digest food in the stomach and then you allow these small particles to go to the small intestine, then you're going to start having a higher concentration of particles there. Okay, so osmolarity is going to increase. That's what hyperosmotic means. This would become a hyperosmotic environment because you are adding particles, right? You're going to have some hydrochloric acid because the walls of the stomach are going to secrete hydrochloric acid, and the hydrochloric acid is going to be here, and the hydrochloric acid is a very small molecule, so you're gonna send hydrochloric acid to the small intestine. And you're gonna have fat. You're gonna have other things, okay? But for the small intestine, fat is particularly important. So the receptors of the small intestine, whenever this food starts going to the small intestine, they are going to detect all these things, right? So you have first an empty small intestine, then the osmolar receptors, they're going to read 0, 0, 0, 0, 0. Whenever the food starts to go to the small intestine, then the osmolar receptors are going to start reading something larger than 0, right? All this information is going to be sent to the central nervous system, and now the central nervous system interprets all this information. Hey, the osmoreceptors are telling me that the concentrations of particles are larger than 0. The acid receptors are telling me that the concentration of hydrochloric acid is higher than 0. The concentration of fat is higher than 0. What do you deduce of these 3 separate pieces of information? Well, that will only happen only if there is food in the small intestine, right? So then the central nervous system has to do something. Now, if the central nervous system, remember, green arrows mean activation, red arrows mean inhibition, okay? So let's consider this situation. Let's suppose that the central nervous system activates the sympathetic nervous system. Now you know that whenever the sympathetic nervous system is activated, this means that immediately the parasympathetic nervous system is inactivated, right? So remember that the sympathetic nervous system, we activate the sympathetic nervous system to elicit the fight or flight response, right? And when we elicit the fight or flight response is when you are nervous, when you are in a situation of stress. So you want to stop processes that are not immediately necessary. And digestion is not one of is one of these processes, actually. So you can wait to digest things when you are confronted with a situation that might be a matter of life and death. All right? So if you activate the sympathetic nervous system, what's going to happen? You are going to reduce stomach motility. You're going to stop this process. You have inactivated the parasympathetic nervous system, so the parasympathetic nervous system cannot do anything. The parasympathetic nervous system, actually, when it's activated, it will stimulate stomach motility, right? It stimulates digestion. So in this situation, you cannot do that. The enteric nervous system is kind of weird. So I told you that the enteric nervous system, I told you that last week, I guess. It works almost independently of the central nervous system. So you get some messages from the central nervous system, but the enteric nervous system can make its own decisions sometimes. And the enteric nervous system, when it's activated, always stimulates digestion, okay? Now, the thing here is that think about what happens when you are nervous or when you are confronted to a stressful situation. So you have problems in the digestive system, right? So I don't know, I guess everybody has experienced that. You are not really hungry, your digestions are not good, and it's because you shut down digestion, all right? You want to put your energy and your resources into something else. You perceive that there is a situation that can be dangerous potentially, okay? In which you might need to elicit the fight or flight response, and then you need to put all your resources ready, available to confront that situation, okay? You don't want to waste resources into things that you can do later. Okay, that's my bad. There was another slide showing what happens when you do the opposite. When you activate the parasympathetic nervous system, then you activate the stomach motility. But well, I guess that if you understand what the parasympathetic and the sympathetic nervous systems do, yeah.
Is there ever a time where like the central nervous system will like say stop something, but then the enteric will decide its own and say no, I'm going to keep doing it?
What, the enteric nervous system?
Yeah, because you said it could make it.
So okay, here what happens is that the stomach or the small intestine and the esophagus, so they don't stop completely their activity. There are always going to be contractions of the stomach and the small intestine. Actually, even when you don't have food in your stomach and when you don't have food in your small intestine, there are contractions going on. The intensity of the contractions is going, and the frequency is going to be much lower than when you have food in your digestive tube, okay? And there are contractions all the time because of the enteric nervous system. And the central nervous system doesn't have absolute control over the enteric nervous system. The enteric nervous system is going to do its thing. The thing of the enteric nervous system is basically controlled motility of the digestive tube and secretion. Also, the secretion is The central nervous system has more control over that, particularly for some things like hydrochloric acid. Okay. But with motility, there is always going to be some baseline motility, right? So there are going to be always like contractions of a small intensity, okay, low frequency. And then whenever you have food in the stomach, the small intestine, you are going to increase the frequency of the contractions, increase the strength of the contractions, all right? And that's going to be controlled by both enteric and the central nervous system. The central nervous system controls that via the parasympathetic nervous system or the sympathetic, right? But when you activate the sympathetic nervous system, it doesn't mean that you're going to stop completely all the contractions. That never happens, okay? That's why I put this green arrow here. This green arrow is because the enteric nervous system always stimulates the stomach motility. But the central nervous system, even in a situation of stress, it cannot tell the enteric nervous system, okay, stop completely your activity. That's not happening. Okay. Any more questions? So, okay, the other slide that I don't know why it doesn't show up is when you reverse that, so this is green and this is red, then this becomes green, right? Then you activate the stomach motility, right? And then you have a joint effort between the parasympathetic nervous system and the enteric nervous system to produce peristaltic waves in the stomach, right? Now these and these happen at the same time, obviously, all right? Now in this case, you have the same thing, but now instead of glands here, you're gonna have the smooth muscle of the stomach, muscle that forms the muscularis externa, so now the effectors are not glands. So, well, I guess that you can do this. Since these things happen at the same time, so here you have exocrine gland. And then you have another set of effectors, right, which are going to be also controlled by both the parasympathetic and enteric nervous system. And in this case, the effectors, it's going to be a smooth muscle of the muscularis externa. Right? And then the response is going to be the production of peristaltic waves. Right? Peristalsis. And here the contraction of the smooth muscle of the muscularis externa obviously doesn't happen in the whole muscularis externa, okay? So you're going to contract regions of the stomach, okay? Contract and relax, so to produce a wave of contraction, okay? Is that clear? Now the overall response of this is digestion of food, right? In the stomach. Any questions about this? Okay, so this is what happens in the stomach. Now the food is in the small intestine, all right? So now we broke down the food, yeah.
So when the sympathetic nervous system is activated, the stomach motility stops?
It doesn't stop completely, but it's reduced. So then, okay, the peristaltic waves in the stomach, they are produced, they are coordinated, let's say, by the enteric nervous system, mainly, right? Now, for example, I don't know if you had breakfast or not, but probably your stomach is empty. This doesn't mean that there is nothing, there is no movement in your stomach. There is. And this movement, but it's, you have waves of contraction, but they are like very, very weak, and the frequency is low, okay? But still, you have waves of contraction. Those are controlled by the enteric nervous system, which is always working, okay? Now, you are relaxed now, I hope. And if I don't know, somebody gives you some food, then you're gonna eat that food, and then since you are relaxed and you are going to digest some food, you're going to activate your parasympathetic nervous system, and then the activation of the parasympathetic nervous system will increase stomach motility, will stimulate the stomach motility. Now, the waves of contraction are going to be more frequent and stronger, right? Because you have to digest all this food. If you are stressed, let's say that you are like, I tell everybody, okay, that we're gonna have a quiz, surprise. Then you're gonna be stressed, and then if you need to eat something, then probably you're not gonna have a very good digestion, because the central nervous system, now you are in a stressful situation, activates the sympathetic nervous system, which inhibits stomach motility. You're still going to have some motility, all right? But probably digestion is not going to be as good as if you are relaxed, right? Is that clear? I mean, more than the details of the system, so I want you to understand this, the big picture, right? And then you can always add details. So I want you to see this as, so I want you to understand the big picture, okay? So I present you diagrams that show kind of the big picture. And then what we're doing all the time is like zoom in, okay? And then zoom out, and so on. So for example, when I was talking about the stomach secretion, so we focused for a couple of minutes on the secretion of hydrochloric acid, which happens at the cellular level. This is what happens. This is what we do to secrete hydrochloric acid. So this is zooming in only here, okay? But once you understand exactly how this happens, then we're gonna zoom out again to see why this happens in the context of homeostatic control mechanisms, okay? And you never, try not to lose perspective, all right? So then we can focus on another element. So we zoom into another element. But then after we have done that, then we are gonna zoom out. And then we're gonna zoom in on another thing, and so on, right? All right, so now we broke down these food items into components, right? These are much more smaller particles than, much smaller particles than whatever you bring into your stomach in the first place, obviously. But there are still some things that need further digestion, okay? So the pyloric sphincter, the orifice, is like pretty tiny, but still you can get like very large molecules that you need to further break down, so for the small intestine to absorb all these things. So, although most of the digestion has happened already in the stomach, you still need to do some more digestion. You still need to break down these things even more. Now, when the food leaves the stomach, it enters the small intestine, and the first section of the intestine is small. It's like 20 centimeters or so. And this first section of the intestine is the duodenum. But under a physiological point of view, it's like it's a tiny portion of the small intestine, but it's actually super interesting. There are lots of things happening here, and I'm going to explain some detail what's going on. So here you have the duodenum represented here. The stomach would be here, but it's removed, okay? So it's cut here and removed. So you can see other structures, right? Now, this thing here is the pancreas. The pancreas is a gland, And the pancreas is formed by, the structure of the pancreas is somewhat complicated, not too much, but. So the pancreas, if you do a cross section of the pancreas, okay, and then you look at, so you do a section like this. And then you take a look at a tiny portion of the pancreas under the microscope, you are going to see cells that form structures like this. Let me see if I can. Do you know the trivia, the game? You have to collect all these wedges, right? So those are going to be cells that look like these wedges of the trivia, and then when you put all of them, like you order them like this, then you're going to have a structure like that, and in the center you're going to have a canal, a duct, right? And you're going to have lots of these structures that are going to be the main structure of the pancreas, right? So cells that surround a tiny canal, right? And then embedded into these structures here, you're gonna have cells that are not ordered at all, so they do something like that. They are oriented like this. Not oriented, but they are structured like that. In the middle of these cells, you're gonna have lots of blood vessels, capillaries. Right? And then you're gonna have those surrounded by these structures here, okay? That would be the structure of the pancreas. And these structures here, collectively, they form what we call the exocrine pancreas. And these things here, so collectively, well, each one of these structures is called an islet of Langerhans, but all the islets of Langerhans in the pancreas, they form what we call the endocrine pancreas. All right, so the pancreas is an organ, it's a gland. Since it's a gland, its main function is to secrete things. And it has two components, the exocrine pancreas, which is going to secrete things to the outside of the body, right? And the endocrine pancreas, which is going to secrete hormones to the blood vessels. Remember that last time we talked about the difference between endocrine and exocrine glands? Now, the exocrine pancreas is going to secrete basically digestive enzymes. And the endocrine pancreas is going to secrete hormones. And you have heard about a hormone called insulin, right? The insulin, for example, is secreted by the endocrine pancreas. Glucagon is another hormone secreted by the endocrine pancreas. There are a couple more hormones that are secreted by the endocrine pancreas. All right? Now, you can see that each one of these structures is formed by these cells that are going to secrete the digestive enzymes, and then they put the digestive enzymes into these small ducts, and you have like thousands of these structures in the pancreas, and all the digestive enzymes are going to travel through these ducts. Then at some point these two, they are going to converge with this one, with that one, and then everybody, all these small tiny ducts, they are going to converge into larger tubes, until all of them converge into this big duct, which we call pancreatic duct. Okay? So that would be like, I don't know, roads, a system of roads or whatever. Or a river, yeah? So you have a really small stream, and many small streams are going to bring water to a larger stream. And then many larger streams, they will get the water into a small river, and maybe lots of small rivers are going to be affluents of the big river, right? And the big river actually ends up in the sea, right? So that would be pretty much the same. The big river, so to speak, would be the pancreatic duct. And the sea is the duodenum. Okay? Now, we have the pancreas secreting digestive enzymes and hormones. And we have another big gland that is part of the digestive system. And this other big gland is the liver. And the liver is not here, so we remove the liver as well. So we remove the stomach and the liver, okay? But the liver is formed by cells that are called hepatocytes. And these cells are going to produce the bile. So the hepatocytes, they have lots of functions, and the liver can work as a gland, right? So it can secrete things. The bile is one of the things that the liver secretes. So you can see here that you have bile ducts that come from the different lobes of the liver. So this means that lots of hepatocytes are going to secrete bile, and this is going to be secreted into a tiny canal. And then other hepatocytes are going to secrete bile, and this goes to another tiny canal, and then they're going to converge, okay? And then eventually you're going to have larger and larger ducts that are collecting all the bile that the hepatocytes are secreting, okay? Then eventually you get the bile stored in the gallbladder. The gallbladder doesn't secrete bile, okay? Just stores bile. And the gallbladder is like a bag, okay? And this bag is hollow. Then the bile is going to be accumulated there. And the walls of the gallbladder, they have a structure that is very similar to the wall of the gastrointestinal tract. So there is going to be an epithelium, connective tissue, and then a layer of smooth muscle. So what do you think will happen when you contract the smooth muscle of the gallbladder? The bile is going to be pushed out, right? Okay, so here you have this anatomical structure. So the bile ducts, the bile secreted by the liver goes to the gallbladder, and then you can see that there is a tube that comes out of the gallbladder, and it merges with the pancreatic duct, and then there is a small duct here that discharges to the duodenum. Okay, so you're gonna have bile ducts. So the liver is going to secrete bile, which is collected in the bile ducts, right? Then it's going to be the bile is going to be stored in the gallbladder. And then you're going to have another duct whenever the gallbladder contracts. Then the bile is going to go through here. And here you have the pancreas. Pancreas is going to secrete digestive enzymes. And hormones. The digestive enzymes are going to be secreted by the exocrine pancreas. The hormones are going to be secreted by the endocrine pancreas. But you know that the hormones are secreted into blood vessels. So we're going to have blood vessels here. And the hormones are going to be secreted to the blood vessels, right? And the digestive enzymes, they are going to be secreted into the pancreatic duct, right? So they're going travel that way, and then eventually these two tubes, they're going to merge. The bile duct and the pancreatic duct, they merge. So then the bile is going to be secreted at the same time as the digestive enzymes. And where is that going to be secreted? To the duodenum. But there is a sphincter here. We call that the sphincter of Oddi. Right? So, the sphincter of Oddi, a sphincter is a ring of smooth muscle, okay? Forms kind of like a gate. And sphincters are closed, usually. So, if you want to open them, then you need to do something. Okay? So this means that whenever we're not digesting anything, whenever the duodenum is empty, the sphincter of Oddi is closed, and then the bile and the digestive enzymes are not going to be able to move to the small intestine, right? That makes total sense, because if you don't have any food to digest, and you keep adding bile and digestive enzymes into an empty duodenum, you're not digesting anything, but what you're doing is wasting energy and resources, right? So you need to discharge the bile and the digestive enzymes only whenever there is food in the duodenum, right? Is that clear? Yeah.
So after the bile goes through the common bile duct, where does it go?
So the bile is continuously produced by the liver, and it's stored in the gallbladder, right? And then whenever we need to release the bile, then the bile will go to the common bile duct, and the sphincter of Oddi will be open. We're gonna see how this works in a second, right? Any more questions? Is that clear? I'm assuming that you're familiar, or at least that this is not the first time that you see the anatomy of the pancreas and the liver. But I don't know, maybe I'm wrong just on that. If you have questions about the anatomy or something, then let me know and we can meet sometime. Okay, so how do we control these secretions? So we have the pancreas. Let's start with the pancreas, all right? This is a drawing of pretty much what I did there, but that's not a cross section. That's different. And you can see that here you have the exocrine cells that are forming a structure like surrounding a canal, right? A duct, a small tube, and here you only have one of each represented, but imagine thousands of those. And in between the exocrine cells, you have the endocrine cells of the pancreas. Those form these structures that are called islets of Langerfeld. But those are going to secrete hormones. So these hormones are going to go to the blood vessels. Okay? Now, those secrete digestive enzymes. And here you have how it works. This is the endocrine pancreas. This is the exocrine pancreas, but I cannot draw like two different organs because those components are in the same organ but mixed. Yeah. So the exocrine pancreas produces enzyme precursors. So remember when we were talking about the secretions of the stomach, that I told you that the cells of the stomach, they produce pepsinogen, and then you need the secretion of hydrochloric acid to break pepsinogen into pepsin. Pepsinogen is inactive, and that's good because you cannot produce an active enzyme that is going to digest proteins, because if you're a cell and you produce pepsin directly, then pepsin is going to eat your own proteins. So you need to secrete first an inactive form of the enzyme, get rid of it, and then something is going to be outside that will activate that enzyme, all right? So the kinds of digestive enzymes that the pancreas secretes break down pretty much everything. So the pancreas secretes peptidases, which are enzymes that break down proteins. Trypsin, proteases, all those break down proteins. They also secrete lipases, which break down lipids. They secrete enzymes that break down carbs. Enzymes that break down nucleic acids. So pretty much all these cells, what they do is to secrete enzymes that are going to break down pretty much all the food items that you can imagine. They need to secrete these enzymes in an inactive form, okay? Because if they secrete the active enzymes, so these cells will die because they are going to be digested by the very enzymes that they are themselves producing, all right? So collectively, these enzyme precursors, they are called zymogens, right? Don't worry. So when you see these words, zymogens, this means like enzyme precursors, okay? Not a specific enzyme, but pretty much all the collection of enzyme precursors. Now, interestingly, the duct cells, they secrete bicarbonate. So these are cells that form these tubules, okay? But they are not just passive, they're not just forming like the tube. They also secrete things. They secrete bicarbonate in this case, okay? Not enzymes, bicarbonate. And here you have the cells of the endocrine pancreas that produce insulin and glucagon among other hormones, but those go to the blood vessels, and we will see what these two do next week. Okay? Why do you think we secrete bicarbonate? What do you think bicarbonate will do?
Absorb?
Absorb?
No, just kidding. I don't know.
What do you think bicarbonate will do? We'll see that in a minute. How these cells secrete all these things, enzyme precursors, bicarbonate, and all these things? Well, you have mechanisms of transport of molecules. Bicarbonate is pretty much this. When you mix this with water, then you get bicarbonate. Secondary active transport, right? To secrete bicarbonate, these cells, if they want to secrete bicarbonate, they need to pump to do some kind of primary active transport to generate an electrochemical gradient. Otherwise, this mechanism cannot work. Okay. Now, that's what's going on in the pancreas. What's going on in the liver? So the cells of the liver are called hepatocytes. Hepatos means liver in Greek. Cyte means cell. Hepatocytes literally mean cells of the liver, okay? And the hepatocytes secrete bile, and bile is a substance formed by a mixture of different things. The most important are bile salts, cholesterol, and bile pigments. These pigments, they also have some functions, but we're not going to extend on that. But this is what gives, the bile is what gives, the bile is kind of greenish, and it's what gives the brown color to feces. So without bile, your feces wouldn't be brown. It would have like different colors. But it's because every time we secrete bile, we recycle the bile salts, but actually we lose all the other components. The ducts where the hepatocytes secrete the bile too, they're called bile canaliculi. Canaliculi means like small canals. Don't worry about these names. I'm just interested in you knowing that the hepatocytes secrete the bile. The bile is a mixture of things, and the bile is collected in the gallbladder until we use it. Okay. All right, and these things are going to have a role in digestion, right? So remember that I told you that when the food comes from the stomach to the duodenum, many things are already digested, but you still have like some things that need to be completely digested. And this is why we secrete the bile and the digestive enzymes of the pancreas. So the digestive enzymes of the pancreas are going to break down pretty much everything. The bile is going to be used to break down lipids, right? Okay, so here you have how do we control the pancreas and the bile secretion. And here you have all the effectors that are going to be involved in that. So those are different organs. You have the small intestine, the sphincter of body, remember this thing that opens or closes, which is normally closed. Okay. The gallbladder, where we store bile, the bile ducts, the hepatocytes, which are the cells that produce bile, and the two components of the pancreas, the endocrine and the exocrine pancreas. Now, the stimulus is going to be the presence of food in the small intestine, right? So you have, here I represented fat, glucose, and hydrochloric acid. Remember that whenever we send the food digested from the stomach, you are going to have some amount of hydrochloric acid no matter what you do, because the hydrochloric acid is a very small molecule and it's going to cross the pyloric sphincter. Hydrochloric acid, theoretically, is going to be pretty bad for your small intestine. For the stomach, it's all right because the stomach secretes a huge layer of mucus. But the main function of the small intestine is to absorb things. If you have a huge layer of mucus on top of your cells, then it's going to be very difficult for you to absorb anything, right? So you're not interested in having a really thick layer of mucus. The downside is that you're not going to be protected against hydrochloric acid, right? So we have a trick to get rid of the hydrochloric acid. And that's what I will explain. Okay, so the small intestine, the duodenum, it's going to respond to the presence of fat, the presence of hydrochloric acid, and the presence of glucose, right? Now, we have cells of the small intestine that are actually going to be endocrine glands, right? And these endocrine glands are going to secrete hormones. Therefore, these things are going to be secreted to the blood vessels, okay? Not to the lumen of the small intestine. So these things are going to be secreted to the blood vessels. And we have this thing called cholecystokinin. Chole is related with bile. Cysto means like a bag in Latin. Kinin, movement. Cholecystokinin is a hormone that stimulates the movement of a bag that collects bile. The bag that collects bile is the gallbladder, right? The presence of hydrochloric acid in the small intestine stimulates the small intestine to produce secretin. And the presence of glucose in the small intestine stimulates the small intestine to secrete this thing that is called glucose-dependent insulinotropic peptide. Peptide means a small protein. Insulinotropic means like producing insulin. And glucose-dependent means exactly that. Okay, so these three things are hormones that are going to be secreted to the blood vessels, and the blood vessels eventually they're going to bring these hormones to the target organs. And it turns out that you're going to have different target organs for these hormones. But before we do that, so remember that what the hepatocytes do is to secrete bile salts. The exocrine pancreas secretes bicarbonate and enzymes, and the endocrine pancreas secretes insulin, right? Now, here you have the elements of this homeostatic control mechanism. Let's start with what each one of these hormones does. Glucose-dependent insulinotropic peptide. Well, this thing is going to stimulate the endocrine pancreas, this is why you have a green arrow, to secrete insulin. Okay? So, the endocrine pancreas is going to secrete insulin whenever this thing is in the blood. And when is this thing in the blood? Whenever there is glucose in the small intestine. Do you know what insulin does? What's the main function of insulin?
Is it to lower your blood sugar?
Yeah, so why, whenever you have insulin, you lower the concentration of glucose in the blood because insulin tells cells to take the glucose. Okay? Why do you want your cells to take glucose? I think the answer is clear, no? What do your cells do with glucose?
Create energy.
Produce energy. So the small intestine absorbs the glucose, right? And then the glucose is distributed via the blood vessels to all the cells in the body, and the cells, they need to take the glucose to produce energy. So one of the signals that cells use to take the glucose is the presence of insulin, but insulin needs to be secreted, in this case by the endocrine pancreas, and the signal that the endocrine pancreas uses to secrete insulin is the presence of this thing in the blood. And this thing won't be in the blood unless there is glucose in the small intestine. Do you see how, what would be the sequence, right? Okay. So far that makes sense, I hope. Secreting, what it does is to stimulate the bile ducts. The bile ducts also have cells that also secrete bicarbonate. And remember that the exocrine pancreas, the cells of the ducts in the pancreas, they also secrete bicarbonate. So, why are we interested in secreting bicarbonate whenever there is hydrochloric acid in the small intestine? That's chemistry, by the way. So, hydrochloric acid is an acid, strong acid. Bicarbonate is what kind of, it's an acid? It's a base. What happens when you mix an acid and a base?
You get salt and water.
Salt and water. Salts, usually they are harmless. So, how are you going to get rid of hydrochloric acid? Hydrochloric acid is very dangerous in the small intestine. In the stomach, it doesn't do any damage, but in the small intestine, it's very dangerous because it can eat the walls of your small intestine. The small intestine the walls are relatively thin, and they're not protected by mucus. Well, so what the bile ducts and the ducts of the exocrine pancreas do is to secrete bicarbonate, which is going to react with the hydrochloric acid, producing salt and water. So now you are safe. Now the small intestine is safe, right? The bile salts, so the hepatocytes are going to secrete bile salts, and actually this is an interesting thing. So the presence of bile salts in the liver stimulates the hepatocytes to secrete more bile salts. That would be an example of a positive or a negative feedback loop. Positive, because you have, in this mini loop, the stimulus would be the presence of bile salts, and the response would be the production of more bile salts. So you go with the stimulus. Right? Cholecystokinin, what it does is to stimulate the contractions of the smooth muscles of the gallbladder. When you contract the smooth muscle of the gallbladder, you push the contents of the gallbladder away, right? The gallbladder stores bile, so whenever you contract this, then the bile goes from the gallbladder to the duodenum, right? It inhibits the contraction of the smooth muscles of the sphincter of Oddi. When you inhibit the contraction of a muscle, then what you are producing is the relaxation of the muscle, right? So basically what cholecystokinin does is to relax the sphincter of Oddi. I'm sure that if you think a bit about it, you know what happens when you relax the sphincters, right? So the things that you're trying to contain, you cannot contain. So when the cholecystokinin relaxes the sphincter of Oddi, what's going to happen with the sphincter of Oddi? It's going to be relaxed, it opens. And then all the contents from the gallbladder and the pancreatic duct are going to be discharged in the duodenum. What are these contents? Bile and pancreatic enzymes. Is it good that we discharge them? Well, it's not good if there is no food in the duodenum, but if there is food, we want this food to mix with the bile and the pancreatic enzymes, right? To finish digesting. The cholecystokinin also stimulates the exocrine pancreas to produce enzymes, all right? Is that clear? So again, you can put that, well, I erased it, but if you do receptors, afferent pathways, central nervous system, blah, 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 you have multiple effectors, multiple levels of response, all right? But the overall response is, what do you think the overall response is? Digestion of food in the duodenum, right? You still have particles that are going to be really small, but you still need to digest them further. And you're gonna use basically the enzymes and the bile to finish digestion, all right? Do you understand this? I'm interested in you understanding the big picture again. So I'm not going to ask specific things about this, and I don't want you to memorize all this. So I want you to understand what's going on here. Because I chose these hormones, but obviously there are many more hormones and many more things involved, okay? Make sure that you understand this, all right? Any questions? Good. So then wait for my email with the comments on your outline, all right?